Hello, everyone, and welcome to today's live broadcast, how LV and AAV transfection platforms will shape the future of gene therapy manufacturing. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Mirus. For more information about our sponsor, please visit their site at mirusbio.com. Now, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking the Ask a Question box located on the far left of your screen. I'd now like to introduce our presenter, Leisha Kopp, Application Scientist at Mirus Bio. For a complete biography on our presenter, please visit the Biography tab at the top of your screen. Leisha, you may now begin your presentation.
Thank you for the introduction, and thanks to everyone for tuning into our webinar on how LV and AAV transfection platforms will shape the future of gene therapy manufacturing. At Mirus, we strive to provide turnkey tools and fantastic scientific support, always with the goal of enabling researchers around the world as they seek to understand and improve the human condition. During this presentation, I will provide a brief overview of Mirus Bio and our core technologies. Then we will discuss the TransIT-VirusGEN products that we've developed for lentivirus and adeno-associated virus production. We will cover the critical parameters Mirus used to develop our LV and AAV kits, as well as some key considerations for choosing TransIT-VirusGEN for your viral vector platforms, including the flexibility to use whichever HEC293 cells, vector sets, and culture media you'd like, how high titers lead to fewer production runs to generate sufficient doses for large patient populations, and how Mirus and VirusGEN will support you from R&D all the way through commercialization. Following this presentation, we have a couple quick polling questions for you, and then we will open the floor for Q&A. Any and all questions are welcome, so please don't be shy. I do look forward to the discussion. Mirus Bio was formed in the mid-1990s by 3 researchers at the University of Wisconsin-Madison with a passion for gene delivery, both in vivo and in vitro. TransIT-LT1, the first serum-compatible DNA transfection reagent on the market, was launched in 1996 and is still one of our most widely used products. Shortly after LT1, Mirus also commercialized reagents for small and large RNA delivery. In addition to chemical transfection reagents, Mirus offers solutions for electroporation with the Ingenio electroporation solution and EasyParator electroporation instruments. More application-specific products included the TransIT Pro transfection reagent and the Trogo high-yield expression system for protein production. Our TransIT X2 dynamic delivery system, introduced in 2013, is perhaps the most broad-spectrum chemical reagent we offer, as it's designed for both DNA and small RNA delivery and performs well with a wide variety of cell types and applications, including gene expression, RNAi, and CRISPR-Cas genome editing. TransIT-VirusGEN reagents that we'll talk about today was first introduced in 2017 as research grade. TransIT-VirusGEN Select, which is designed for preclinical and early phase clinical production, was introduced last year. TransIT-VirusGEN GMP and our VirusGEN LV and AAV kits will be available in early 2021, and those are the products we'll focus on today. In conversations with researchers, we often get the question, what makes Mirus reagents different? So I'd like to take a moment to address it. While many transfection formulations utilize a cationic polymer like PEI or liposomes like lipofectamine, Mirus formulations are non-liposomal mixtures of both cationic lipids and polymers, which promote effective complex formation to a broad spectrum of cells. Our formulations also tend to be less toxic than liposomal formulations because they're less disruptive to the cell membrane. And unlike PEI-based formulations, Mirus reagents are effective for delivery of multiple nucleic acids to a wide range of cell types for many different applications. I'd also like to add that Mirus strives to provide the best support possible for our customers. Our scientific team is comprised of world-class chemists and biologists committed to finding those perfect formulations and protocols to streamline and improve transfection-mediated applications. Additionally, we offer a technical support team of R&D and application scientists that work closely with the reagents you're interested in, so when you reach out to us via phone, email, or web chat, you immediately reach an expert that can help you address your questions on the spot. To summarize, Mirus Bio is a nucleic acid delivery company dedicated to enabling research with products for general DNA delivery and DNA-RNA co-delivery, products for specific applications such as viral vector production, and products that utilize alternate strategies for transfection, such as electroporation, all with the goal of supporting any delivery method, any nucleic acid, for all cell biology applications. In our 25-year journey, we've made some friends along the way. Mirus has over 30 trusted distribution partners supporting over 40 countries around the world in all major markets. If interested, you can find our list of international distributors at mirusbio.com/contact/distributors. We also offer international direct ordering to countries for which we do not currently have a distributor. Now that you know a bit more about Mirus, let's dive into our discussion on the TransIT-VirusGEN reagent as well as the VirusGEN kits for LV and AAV production. I'm sure it's no surprise that supporting cell and gene therapies was top of mind when developing a new transfection reagent for LV and AAV production. As we all know, lentivirus is often a critical component of cell therapy, as shown in this example, where patient cells such as T lymphocytes or NK cells are harvested and then genetically engineered to express a protein such as a chimeric antigen receptor or a CAR that better enables the cell to detect and eliminate cancer once expanded and re-administered into the patient's body. Adeno-associated virus is commonly used in gene therapy, where the virus itself is the therapeutic as it contains a corrected copy of a gene which can be directly administered to the patient. Currently, there are 3 cell therapies and 2 gene therapies approved by the FDA. In 2017, Novartis' Kymriah, which is designed to treat acute lymphoblastic leukemia, and Kymriah, which is designed to treat non-Hodgkin lymphoma, were introduced. Very recently, in July of 2020, the FDA approved Kymriah for treating relapsed or refractory mantle cell lymphoma. On the gene therapy side, in 2017, Spark Therapeutics launched Luxturna, which is directed at correcting inherited retinal disease due to mutations in RPE65. In 2019, Avexis introduced Zolgensma, an AAV-based therapy designed to treat spinal muscular atrophy. Now, each of these therapeutics is groundbreaking and life-changing, which is why there's so much effort in this field, which also guarantees that there will be many more to come. According to data recently published in Bioprocess International, there are currently more than 1,000 cell and gene therapies in development. This figure shows the projected cell and gene therapy clinical trials through 2026, with actual numbers dating back to 2010. The real take-home here is that these therapies are growing at an unprecedented rate, and it appears that we are only at the beginning of this curve in 2020. This figure suggests that there will be between 10 to 12,000 cell and gene therapy clinical trials by 2026. The graph at right breaks down the current product candidate pipeline and highlights that lentivirus and AAV are the predominant viral vectors of choice for these therapeutics. When designing a reagent to support cell and gene therapy development, we had several goals in mind. First, we knew that we needed a reagent formulation that supported delivery of multiple plasmid DNAs to HEK cells, which is the basis for LV and AAV generation via transient transfection. Beyond that, we wanted to develop a reagent that was best in class, so to speak, for not just LV or AAV, but for both. Secondly, we screened libraries of our lipids and polymers for a combination that promoted effective complex formation for both adherent and suspension cell types, as the formulation would then be able to support large-scale LV and AAV production in formats such as the iCELLis and roller bottles, as well as stirred tank bioreactors. And last but not least, we knew we needed a formulation that supported reproducible and scalable virus production. Ultimately, our goal was to simplify the viral vector development process by delivering a formulation that yielded more functional virus per transfection to streamline processes and reduce production costs, and the TransIT-VirusGEN is able to do those things. The features of our TransIT-VirusGEN formulation include superior performance, cell line and platform flexibility, and batch-to-batch reliability. More data on those points to come. In addition to a functional QC, VirusGEN Select, shown here, includes release testing and quality documentation to streamline the process of raw material qualification to ensure a seamless transition from discovery through large-scale manufacturing. TransIT-VirusGEN is chemically defined and animal origin-free, and there's no commercial license required for R&D or manufacturing use. PEI-based reagents are the most commonly used formulations in viral vector manufacturing, which is really what sets the bar for reagent development at Mirus. This data, generated in suspension 293F cells grown in freestyle F17 media, demonstrates that VirusGEN outperforms both 25 kilodalton PEI and PEI Pro from Polyplus by generating up to 10-fold higher functional titers for lentivirus and up to 4-fold higher functional titers for AAV production. VirusGEN, as demonstrated in this figure, also strongly outperforms PEI formulations in adherent 293 T cells grown and transfected in DMAM plus 10% FBS. If you'd like more details about these experiments, please visit our select page at www.mirusbio.com/select. In these next few sections, we'll look more closely at critical workflow parameters that impact lentivirus and AAV expression levels, as well as how these factors guided development of our LV and AAV kits and recommended protocols. This figure depicts the process we followed when developing the VirusGEN LV and AAV kits. The first step was scouring the literature as a starting point for development of candidate compounds. We then tested each of these potential enhancers for performance in our established lentivirus and AAV workflows with TransIT-VirusGEN. With the most promising candidates, we investigated other critical parameters, including DNA amount and cell density at transfection for both adherent and suspension cells. And with our final LV and AAV systems, we also compared expression levels with various sets of plasmids, various serum-free cell culture medias, as well as different suspension 293 cell types. The first critical component we will explore are the impacts of total DNA and cell density at the time of transfection. These figures show a DNA titration from 1 to 2 micrograms of total DNA per mil of culture for both LV on the left and AAV on the right. We found that lentivirus production without the LV enhancer, the black bars, is higher when using 1.5 to 2 micrograms of total DNA. However, only 1 microgram of DNA is needed when using the LV enhancer. This data also shows that the LV enhancer boosts titers 3 to 5-fold over reagent alone conditions. For AAV, we found that 1.5 to 2 micrograms of total DNA per mil of culture is optimal with and without the enhancer, and that the optimal DNA amount will vary depending on the serotype and vector sets used. As shown by the red bars at right, the enhancer doubles AAV production in all conditions. This does bring up an important point for platform development, though, as the total DNA requirement for optimal expression may depend on the interactions of several components. For this reason, we highly recommend running a DNA titration with your final vectors, cell, and medias to determine the lowest DNA amount you can use for robust expression. The cell density at transfection is another commonly explored parameter. For lentivirus and AAV production with TransIT-VirusGEN alone, meaning without the enhancer, 2 million cells per mil at transfection was best. In contrast, significantly higher titers are observed when using the LV enhancer, the red bars, at 4 million cells per mil. For AAV, we see equivalently high titers at 2 and 3 million cells per mil, both with and without the enhancer. Again, it's a good idea to do a cell density titration with your final cells, gene of interest containing vectors and media to determine what is optimal. Based on the data we just covered for DNA and cell density at transfection, here is the final protocol for the VirusGEN LV kit. The day before transfection, cells should be split to ensure that they are rapidly dividing at the time of transfection. On the day of transfection, dilute cells to 4 million cells per mil, form transfection complexes per the protocol by combining 1 microgram total DNA and 3 microliters TransIT-VirusGEN per 1 mil of total culture, incubate 10 to 15 minutes, add complexes to the cells, and place cultures at 37 degrees, shaking overnight. 18 to 24 hours post-transfection, add the LV enhancer to the cultures at about 10% of the total culture volume. And 48 hours post-transfection, harvest your lentivirus from the supernatant and assay as desired. For the AAV kit, the upfront work is similar, however cells should be diluted to 2 million cells per mil for transfection rather than 46 per mil. Also, the AAV enhancer is included in the complex formation solution, so there's no need to reopen the culture post-transfection. At 72 hours post-transfection, or whatever time is best per your AAV serotype, harvest and purify or assay as needed. Using the protocols outlined in the previous slide, we again compared the performance of VirusGEN as well as the VirusGEN LV and AAV kits to both 25 kilodalton PEI and PEI Pro in suspension 293F cells and freestyle F17 media. With the revised protocols and enhancers, our improvements over PEI-based methods is another 2 to 3-fold greater than what was achieved with VirusGEN reagent alone. The next critical component we would like to take a closer look at is vector design. Until recently, most of our VirusGEN reagents and LV kit development work was done with 3 different packaging plasmid mixes. The first being the 3rd generation virus-safe lentiviral packaging plasmids from Cell Biolabs, as well as the 2nd and 3rd gen hybrid mission lentivirus packaging mix from Sigma, and the 3rd gen LV Max packaging plasmids from Thermo. Traditionally, we saw highest titers using the mission packaging mix, which isn't unexpected since there are fewer total plasmids to transfect with that 2nd, 3rd gen hybrid system. However, more recently we tested the PALD LV packaging and transfer plasmids from Aldevron, and honestly we were blown away by their performance. Here we see functional LV titers nearing 1 billion TU per ml, which really raises the bar for commercial plasmids and also begs the question, what else does this improve? Do these types of vectors enable the use of less DNA and reagent to get equivalently high titers? In this next experiment, we transfected 293F cells at 4 million cells per mil with total PALD vector DNA ranging from 0.5 micrograms per mil to 2 micrograms per mil with TransIT-VirusGEN and the VirusGEN LV kit. The black bars show titers without the LV enhancer, and we see that while titers are still very high at 1 microgram per mil, they do increase when using more DNA. However, when adding the LV enhancer post-transfection, the highest titers are achieved when using 0.75 to 1 microgram total DNA per mil. This is another nod toward doing a DNA titration with your final system, as less DNA may be required when all components are optimized together. Here we compare performance of the VirusGEN LV kit against PEI Pro and PEI Max from Polysciences. For LV expression using the PALD vectors in XB293 cells growing in XB293 media. Titers for all reagents are very high, however, we still see 3.4-fold higher titers versus PEI Pro and 5.4 higher titers versus PEI Max with the TransIT-VirusGEN LV kit. Over the last few years, we've had conversations with researchers all over the world working to develop LV and AAV suspension platforms. What we've found is that many, if not the majority, of groups are using XB293 or viral production cells from Thermo, and there seems to be a shift away from 293F cells due to concerns with consistency. We also hear many different serum-free medias mentioned, though there are also commonalities there, which leads us to the next critical culture components for LV and AAV expression, cell culture media, and cell type. In this lentivirus expression experiment, we used the PALD packaging vectors from Aldevron and a transfer plasmid expressing GFP to compare performance of the VirusGEN LV kit to PEI Pro and PEI Max. XB293 cells were adapted for several weeks prior to transfection into XB293 expression media, freestyle F17 expression media, LV Max production media, and the balanced CD HEC293 media from Irvine Scientific. Cells were then transfected with 1 microgram total DNA at 4 million cells per mil. We found that the VirusGEN LV kit yielded 3 to 7-fold higher functional titers as compared to PEI Pro and PEI Max. I'd also like to note that we've repeated this experiment several times, and that trend is very consistent. Another trend we see overall is highest LV expression with the balanced CD media and XB293 medias with slightly lower titers consistently observed in free cell F17 and the LV max media. We performed a similar experiment using XB293 cells adapted to the same four medias. However, this time for AAV2 GFP expression using the AAV helper-free system from Agilent Technologies. Transfection complexes were formed with 1.5 micrograms total DNA and 3 microliters VirusGEN reagent per mil in the AAV complex formation solution and enhancer. Results show up to 17-fold increase in AAV titers over the PEI-based formulations when using the VirusGEN AAV kit, and again we see highest overall titers with the balanced CD293 and XB293 medias. For this next experiment, we adapted viral production or VP cells to our suite of four medias and transfected for AAV2 GFP expression. As a control, we concurrently transfected XB293 cells growing in XB293 media. There are a couple noteworthy observations. One, we continue to see great results with the VirusGEN AAV system. Two, titers with VP cells are fairly similar to those obtained with XB293 cells. And three, highest titers are again obtained with the balanced CD media. Another noteworthy point is that expression with our control is at about 42 million TU per ml, which is very close to what we observed at the previous experiment performed 2 to 3 weeks earlier. This demonstrates consistent performance over time, which is always a really good thing to see. Now that we've gone through a slide or two of data, I'd like to highlight considerations for selecting TransIT-VirusGEN for your viral vector platform. Really, it all comes back to this figure here that predicts between 10 to 12,000 cell and gene therapy clinical trials by 2026. If we think about what that really means in terms of production volumes, it's a daunting concept. We'll walk through this a bit more, but it can take hundreds to thousands of liters of AAV to generate a sufficient number of functional viral particles for a single patient dose. If multiplying that by tens of thousands of trials and an even higher number of patients, it begs the question of, can we make enough? As it turns out, there are many people asking that same question. In the last 3 to 4 years, there have been countless announcements by large pharmaceutical companies, CMOs, and CDMOs, such as Fujifilm, Paragon, Lonza, and Brammer Bio, stating that they plan to invest hundreds of millions to billions of dollars in expanding their infrastructure for cell and gene therapy manufacturing. It's likely that much of the added manufacturing space will be devoted to gene therapy and AAV production, given the typical dosage requirements per patient. To explain, the average cell therapy requires between 10 to the 8th and 10 to the 10th viral particles per patient. For cell therapies, a virus is used to transduce cells ex vivo and therefore will never need to contend with the patient's immune response. For gene therapy, the average patient dose requires 10 to the 11th to 10 to the 16th viral particles to be effective. This is because the virus is directly administered to the patient and must affect the entire target organ or system. In addition, the virus will contend with the patient's immune response. If we do the math and translate viral particle requirements to the amount of cell culture required to produce that much virus, it becomes very evident that higher titers are important. So let's do that math. Let's say that a locally administered gene therapy dose requires 3 times 10 to the 11th viral genomes, and a systemic dose requires 5 times 10 to the 15th viral genomes. We get roughly 3 times 10 to the 9th VG per ml from PEI-based production, and assuming even just 4 times higher titers, we get about 1.2 times 10 to the 10th VG per ml with VirusGEN. If we multiply that out, we find that greater than 1,600 liters of AAV culture is needed to generate one systemic patient dose using PEI. When using VirusGEN, that required culture volume per systemic dose drops to just over 400 liters. If we think about what higher titers mean on the production side, it's a significant reduction in material requirements when fewer bioreactor runs and far less media and costly GMP-grade DNA is needed to generate a sufficient amount of virus. It's also less hands-on time, less paperwork, less time in expensive manufacturing suites. And really, in summary, higher titers mean more efficient use of time and materials and an overall reduction in workflow costs. think we've established that higher titers are a good thing. Beyond high titers, what are the other requirements for medical research and pharma groups in the cell and gene therapy space in terms of transfection reagents? First, they need a product that promises consistent high titer virus production. We need support for virus production in both adherent and suspension cells because all formats will be necessary to meet the demand. We need a transfection reagent that will be manufactured under cGMP, and scalable, cost-conscious production processes with a reasonable workflow is critical. At Mirus, we've worked very hard to ensure that VirusGEN reagents and our prototype LV and AAV kits meet those demands. As I mentioned earlier in the talk, VirusGEN is designed to support you from R&D through commercialization. Our research-grade product is configured in vials of up to 1.5 mils and is sterile-filtered and functionally QC'd. TransIT-VirusGEN Select is designed to support preclinical and early-phase clinical trials and is dispensed at 30 ml per bottle. Assays for sterility, bacterial endotoxin, mycoplasma, and formulation identity are performed per USP guidelines on VirusGEN Select and are reflected on the C of A, which we'll see in the next slide. VirusGEN GMP reagent will be available in early 2021, along with the LV and AAV enhancers. VirusGEN GMP will be a 150 ml dispense, which is sufficient for 50 liters of transfection, and will also undergo the same quality release testing as is performed on VirusGEN Select. All VirusGEN reagents are identical in formulation, ready to use, chemically defined, and animal origin free. Documentation provided with TransIT-VirusGEN Select is shown here and can also be found on the TransIT-VirusGEN Select product page, which is shown below. To summarize, the COO and TSC/BSC documents state that TransIT-VirusGEN Select is chemically synthesized in the US and that there is minimal risk of contamination with bovine spongiform encephalopathy, or BSE, or transmissible bovine encephalopathy, or TSE. Our certificate of analysis lists the quality control testing and results used for release of the VirusGEN Select product. As mentioned before, assays for functionality, sterility, endotoxin, and mycoplasma are performed. With VirusGEN, you can expect strong lot-to-lot consistency, as is demonstrated by this data, where we compared LV and AAV expression in 293F cells using 4 unique production lots of TransIT-VirusGEN reagent. VirusGEN is also unaffected by multiple filtrations. In this experiment, we compared LV and AAV expression with an inventory vial of VirusGEN, as well as VirusGEN that had been filtered once, twice, or 3 times through a 0.2 micron filter. As you can see, there is no change in expression levels with additional filtrations. If you're interested in testing either the VirusGEN reagent or the VirusGEN LV or AAV kit for evaluation in your viral vector production platform, we definitely want to enable you to do so. So please visit our sample request portal at mirusbio.com/sample and choose the VirusGEN Select from the large-scale protein or virus production section. Once we receive and qualify your request, we will contact you to verify which sample you'd like. If beta testing the LV or AAV kits, we'll arrange a prep talk with you prior to your evaluation of the kit to walk through the protocol and address any questions that you may have. You'll then receive and evaluate the sample. Please note that we're here to assist with any questions that you have during that time, so don't be shy if you'd like to discuss preliminary results or need to troubleshoot. Following your evaluation, we ask that you send us feedback. You do not need to share any proprietary information. However, we do want to know your thoughts on the protocols and overall performance of VirusGEN compared to your current production system. Going back to the very first slide, the title of our presentation, How Will Optimized LV and AAV Workflows Shape the Future of Cell and Gene Therapy Manufacturing? Well, with products like TransIT-VirusGEN that enable high-titer lentivirus in both suspension and adherent cells for cell therapies, and high-titer AAV production in suspension cells for gene therapies, more therapeutics can be developed and more doses created to treat more people. Ultimately, these transformative technologies enable us all to deliver innovation, empower discovery, and improve life. If you'd like to learn more about the TransIT-VirusGEN reagent or kit, There are a number of resources at your disposal. The TransIT-VirusGEN Select page is shown here, as is our sample request portal. As you can hopefully see, the VirusGEN Select is circled in red. There are also webinars and video tutorials available to you along with frequently asked questions page. You can also download the VirusGEN brochure and documentation from our website if you're interested. To summarize, the TransIT-VirusGEN reagent and VirusGEN LV and AAV kits were designed to offer superior performance, flexibility, reliability, and quality to cell and gene therapy researchers. VirusGEN is also AO-free and requires no commercial license for R&D or manufacturing use. VirusGEN will be available in 3 production grades, all identical in formulation, ready to use, and chemically defined, so one reagent formulation can be used for all stages of virus production. TransIT-VirusGEN RUO is functionally QC'd and comes in vials ranging from 0.3 ml to 1.5 ml, as well as multi-packs of the 1.5 ml configuration. TransIT-VirusGEN Select is designed to support the preclinical and early phase clinical stages and is dispensed in 30 ml per bottle, which is sufficient for a 10-liter transfection. TransIT-VirusGEN RUO and Select are commercially available now and will support viral vector platforms from discovery to early phase clinical trials. The TransIT-VirusGEN GMP reagent, along with the LV and AAV enhancers, will be available in early 2021 and will support late phase clinical trials and commercial viral vector manufacturing. And we are beta testing the VirusGEN LV and AAV enhancers now, so please let us know if that's something you'd like to explore. We sincerely thank you for taking time to learn about Mirus Bio's TransIT-VirusGEN reagent and prototype VirusGEN LV and AAV kits. You're welcome to contact me at any point with questions at the email address listed here, so leisha.kopp@mirusbio.com. You can also contact our tech support team via phone, email, or live chat on our website. We do have a couple polling questions for you now, and then we look forward to further discussion during Q&A.
Okay, so I think everyone can see the survey questions, hopefully. But our first question for you is, what culture medium will you use for your large-scale production process? We want you to select all that are applying. Hopefully everyone had a chance to answer that. Gives you about 10 seconds. Let's see what the results are. All right, LV Max, the balance, and other medias. So we've got a good distribution here. All right, great, thank you. Okay, so then we'll move on to the second survey question. And that is, what is the smallest scale cell culture format that you trust will translate to equivalent large-scale production? And for this, we would like you just to select one answer. All right, it looks like we didn't have a ton of people answer these, but a 1-liter bioreactor. Oh, there we go. Okay, we have more coming in. Excellent. All right, so it looks like people are trusting the bioreactors and then maybe a little bit less the Amber systems. So that is great, very good to see. All right, thanks so much for that.
Thank you, Leisha, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located to the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. All right. So, Leisha, the first question I have for you is how much material to do we receive for beta testing the VirusGEN AAV kit? When can I receive a kit for testing?
Okay, great question. So the material that we will send those that are beta testing the VirusGEN AAV and LV kits will be sufficient for 1 liter of transfection. And so what that essentially means is you'll receive 3 milliliters of the VirusGEN transfection reagent, and then 100 ml bottles of the VirusGEN AAV complex formation solution and enhancer if you're testing the AAV kit. And for the LV kit, it's 2 different 100 ml bottles. One is the complex formation solution, and the other is the LV enhancer that's added 18 to 24 hours post-transfection. So essentially, yes, sufficient material for 1 liter of transfection.
Thank you. So our next question coming up is, how does the amount of DNA required compared to PEI Pro or LV Max kits?
So ultimately, the total amount of DNA required is going to depend on a few things, including the vector sets that are used, the cells that are used, and likely even the media, so that final system that you decide. But that said, we typically recommend 1 microgram of total DNA per mil of culture, whereas other systems like the LV Max system require a higher DNA amount, so typically 2.5 micrograms of DNA per mil. We've also had feedback from customers generating AAV that they have to use about half the total amount of DNA with VirusGEN as they do with PEI-based formulations like PEI Pro. And so again, like I always encourage people to do those titrations of DNA amounts to make sure that they have optimal production levels. But typically with our reagents, in particular, particularly when using enhancers, that 1 microgram per mil is pretty safe.
Awesome. Thank you, Leisha. Our next question is, how do enhancers increase viral production?
That's a good question, and we see that question quite a bit. It is our belief that the enhancers have a stimulatory impact on the cells themselves, which allow them to increase production of the viral components. We know that the enhancers don't impact the transfection part, so they don't increase endocytosis or the ability of the cell to bring in different size transfection complexes, because we've tested addition at various time points. For the LV enhancer, in fact, we add that post-transfection, so definitely not impacting transfection there. But the AAV enhancer, we've tested timing of addition of the enhancer components anywhere from 0 to 24 hours post-transfection, and we see that boost regardless. So it's not transfection that it's impacting; it's the cells that it's impacting.
Thank you. And our next question coming in, what kind of support can you offer if we evaluate your products?
Right. So Mirus really does strive to provide excellent customer service. And so if you're evaluating VirusGEN for a large scale format or one of the kits, so LV or AAV, what we like to do is schedule a prep talk with you. I think we mentioned that in the presentation, before you evaluate. And so we'll walk through the protocol. We'll talk about the different factors that you may want to optimize with your specific sets. We can go through the data that we have. Obviously, we showed quite a bit of data here today, but we have a lot more, right? Our R&D team has generated a lot of data, and we can share that with you, but really we're there to answer any questions that you have and help you troubleshoot if that's needed later on. And the AAV and LV enhancer material is not yet a commercial product, but if you evaluate that and it's something that you want to move forward with, then we'll definitely talk about making sure that you have that to continue developing your large-scale process. So really, I think the take-home is just Mirus is here to support you, so make sure that you utilize us, ask questions. Yeah, I'd say that about sums it up.
Great, thank you. Our next question from one of our audience members, how do you culture the viruses in the suspensions, and what is the incubation time, and what is the protocol of use of suspension media?
Okay, so for producing LV and AAV and suspension cells, the protocol is a little bit different for each virus type, so I guess I'll go individually. For lentivirus, we would recommend doing the transfection, adding the enhancer at 18 to 24 hours post-transfection, and then the harvest is earlier, so it's 48 hours post-transfection, so just a day after you're adding that enhancer to the cells. And to harvest lentivirus, it's you essentially just collect the supername because the virus is leaked out of the cells into the soup, so you'll collect that and purify from there. For AAV, it's going to depend a bit on the serotype that you're working with. So we know that some serotypes can be harvested as early as 48 hours. Some of them are maintained within the cell, some of them are secreted. And so you just have to know the characteristics of the serotype you're working with. But typically, and as our protocol spells it out, we would recommend doing the transfection, incubating at 37 degrees, and then harvesting your AAV at 72 hours post-transfection. And a lot of the AAVs are contained within the cells, and so you'll need to lyse the cells through various methods. It could be freeze-thaw if you're at a smaller scale, or a detergent-based lysis if you're at a larger scale. And then you would harvest according to whichever process that you're using.
Thank you. Our next one coming in, what serotypes have been tested with the VirusGEN AAV kit?
Okay, so at Mirus, we have tested AAV2 primarily. And so a lot of the data that you saw there is the AAV2 GFP from Agilent that we've been using. But we have customers that are testing in numerous other serotypes, including, so AAV2 is common, 5, 6, 8, and 9 are also quite common. And so we've seen folks testing across those, as well as those that have specialized or proprietary capsids. So many different AAV serotypes.
Thank you. I want to thank the audience for all of our questions coming in. The next one is, what are the challenges faced while developing recombinant LV and AAV for gene therapies?
So I think there's many challenges in developing these types of products. From the production side, it's, there's a lot of choices to make, right? You have to choose the right cells, the right media, and find that right platform that gives you the highest possible functional titers for production. And then scaling is another concern that we hear a lot from other PD teams. That's one of the questions, one of the polling questions. What size do you trust for scaling? And so we see people testing a lot in the amber systems, and we see people testing a lot, they'll start in shake flasks and then move into the 1 liter or 2 liter bioreactors. Because they want to know that it scales. If it works great in a small format, is it going to continue to work great? Is it scalable for our GMP process? So there are all of those bits of the workflow to consider. And of course, when it comes to the regulatory side of things, if you're in a GMP process, there is a lot of paperwork. So you need to know that you're qualifying every component that goes into your system. Every process that is used is very heavily documented. And and so the regulatory side is a whole different ballgame, but essentially just a lot. You have to cover a lot of those bases. So I would say from start to finish, a lot of hurdles, but these are being developed rapidly by a lot of people, and so all of these things are being worked out too.
Thank you. Our next question, do you have an assay for detecting residual VirusGEN reagent?
That is something that we are actually working on right now. So we are working with Charles Rivers Laboratories to develop the residual reagent detection assay. And that is something we'll have likely later this fall and certainly available at the time of product launch. So yes, we will have that. And it's something that you'll be able to send your virus to a company, they will run the assay and make sure that it is cleared from the bioproduct.
Okay, we have time for one more question. This viewer says, I am in Sierra working in a veterinary lab. How will I get your products if I need them?
Great question. And so we have distribution partners around the world, and if we don't support any specific country, then we will talk about whether a distributor that is close to you is able to support shipping a product to you, or we can talk about just direct shipping as well. So definitely contact us, and we will talk about all of those options because we want to enable people to test these kits, obviously.
Thank you so much, Leisha. I want to remind our audience that those questions we were unable to answer today and those that come in during the on-demand period will be answered by our speaker via the email address you provided at the time of registration. Thank you again, Leisha. Do you have any final comments for our audience?
Yes, I guess I would just like to thank LabRoots for the opportunity to share our viral vector reagent development journey with everyone that's listening. And of course, Mirus would like to thank everyone that participated today by listening in and asking questions. And thanks in advance to those that listen later to the on-demand webinar. We really appreciate your interest in TransIT-VirusGEN and the enhancer kits. And we truly look forward to working with you and supporting you as you work to develop your own LV and AAV workflows. So thanks again, everyone. We really appreciate your time.
Thank you so much, Leisha. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Again, questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to once again thank Leisha for her time today and her important research. We would also like to thank LabRoots and our sponsor Mirus for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That is all for now. Until next time, thank you, everybody. Goodbye.